we have to check it out that so the story of RBC. We begin from poiesis. So what do you call the RBC production? Production of RBC is called as
Erythropoiesis.
erythropoiesis. The production of RBC is called erythropoiesis. Organ which produce the RBC is called erythropoietic organ. That means poiesis means production. The organ which is in the production of RBC, we say erythropoietic organ. So, who are the erythropoietic organs? Who are the erythropoietic organs? The early embryonic stages, we say yolk sac mesoderm is going to be that erythropoietic organ. In early embryonic stages, the production of blood cells is possible by yolk sac mesoderm. Then after, liver took the role. Liver, some extent, spleen also that took the role. Okay, so the production of RBC, liver and spleen. But from the late fetal stages till after the birth, even that the complete responsibility for the production of blood cells, including our RBC, is red bone marrow. That's what we learned in the previous class. Okay, so yolk sac, mesoderm, liver, spleen, and red bone marrow. Okay, so let's continue the story here. The red bone marrow, production of RBC. So from where it will produce? In the red bone marrow, there are some special cells are present. We call them as hemophoeitic stem cells. The cells are called hemophoeitic stem cell. That hemophoeitic stem cells are going to produce myeloid stem cells. Okay, that myeloid stem cells. So we are talking only about RBC here. That means if you talk about the lymphocytes, we need to go with the lymphoid stem cell. So hemophoeitic stem cell produce myeloid stem cell. These myeloid stem cell produce proerythroblast cells. Proerythroblast cells. These proerythroblast cells form erythroblast cells. Proerythroblast cells form erythroblast cells. Okay. So here that we have some space problem. So that what we do here. Let us type this. Okay. So that make us we'll get some space. Hemophoeitic stem cells. From the hemophoeitic stem cells, that myeloid stem cells are developed. From the hemophoeitic stem cell, myeloid stem cell. The myeloid stem cells develops proerythroblast cells. They develop proerythroblast cells. Proerythroblast cells is going to mature in the form of, that means that next they form erythroblast cells. Erythroblast cells will develop into reticulocyte. Interestingly, till reticulocyte, that we'll find all the cytoorganelle, presence of cytoorganelles you can see here, till reticulocyte. Okay, then after that only, that reticulocytes are even said to be as immature RBC. Reticulocyte, don't be confused with the earlier learned reticular fibers. They are different from here. Okay, so immature RBC, that we say it as immature RBC. Okay, so immature RBC, that it contain all types of cytoorganelle. But we said in case of RBC there is no cytoorganelle. So reticulocyte transforming into RBC means losing all the cytoorganelles and filling the total RBC with the hemoglobin content. That filling the total RBC with the hemoglobin content. Okay, so that was the story of RBC. And after this particular RBC lifespan we have to talk about, because we discussed about the hemoglobin and their components in the earlier class. So that lifespan of RBC is about 120 days. So after that, the completion of lifespan, that 120 days, they die. Where they go? They go into the graveyard of RBC. So which organ of the body is considered as the graveyard of RBC? The spleen. Spleen is considered as a graveyard of RBC. So what happens in the RBC, in the graveyard of RBC with the dead RBC? In the dead RBC, hemoglobin is separated in the form of heme and globin. Globin is a protein. That means dead RBC, the hemoglobin is separated in the spleen in the form of heme and globin. Globin is the protein part that will be recycled. The globin is the protein part. It can be recycled. Then what about the heme? Heme is toxic material. Heme is a toxic material. The toxic heme is going to be sent towards the toxic heme is sent towards detoxified internal organ of the human body. Detoxified organ of the human body. Who is that? Liver. The very important thing to be remembered here. The liver is a detoxifying organ. Any toxic material, for the detoxification, they should reach towards the liver only. Okay. So the for detoxification, the heme will be sent to the liver. So do remember, the globin is used back again, recycling, but we can't use the heme. So the detoxified heme will be sent to the liver. Liver detoxify this in the form of bilirubin and biliverdin. The two pigments we call bilirubin and biliverdin. That means what is bilirubin, biliverdin? You may call it as bilirubin and biliverdin. If you heard before, you may have this kind of pronunciation also. Bilirubin and biliverdin, no matter. That is very important to know. Bilirubin and biliverdin, or bilirubin, biliverdin. What are these? They are simply said to be detoxified parts of the heme. Detoxified heme is said to be in the form of bile pigments. They are in the form of bile pigments. Hope you may understand that color is yellow. That the pigment color is yellow. So that's why I made it with yellow. Okay, so what happens to this bilirubin and biliverdin? Liver produce them actually. The liver produce them. So after production of these things in the liver, they're going to be sent into the alimentary canal. So they are going to be sent through the alimentary canal. That means along with the bile juice from the liver and gallbladder, which are going to send this bile juice, along with the bile juice, these particular bilirubin and biliverdin are going to enter into the alimentary canal. Okay. So that means why they are going to the alimentary canal? What is bilirubin and biliverdin? That bilirubin and biliverdin are the pigments actually the detoxified waste materials. Okay, the detoxified forms of the heme, we have to eliminate them. So what is the way of elimination? There are so many ways for elimination. Some toxic wastes will go through sweat, and carbon dioxide go through the nostrils, the fecal matter through the anus. Likewise, the bilirubin and biliverdin is going to join with the alimentary canal. Okay, is going to join with the alimentary canal, and these pigments from the liver through the bile juice, so they come out through the alimentary canal. This is what happened here. Okay, so that means from the production of RBC, after the death of RBC, in the RBC, whatever the heme is there, that heme is going to be detoxified in the form of bilirubin and biliverdin, and these pigments are going to be eliminated out through the fecal matter of the human being, that is through the rectum and anus. So that's the reason whatever the food materials we eat, no matter that the color of the fecal matter is always is going to be yellow in color. That means that is because of, we say, these pigments, because every day some RBC will die, isn't it? 120 days back, whatever the RBC produced over, they'll die today. So tomorrow also some RBC will be expired over. That heme is going to be by the liver detoxified in the form of bilirubin and biliverdin. If any blockage of these or if any failure of the liver or gallbladder, liver actually produce and gallbladder stores. If any such thing is happened over, this bile, if it is not moving in the way which I am showing, if it is escaped and it seem in the bloodstream, if it is in the bloodstream, you may know that so-called the famous the disease jaundice that makes the appearance of yellow color. Okay, the actual way, every day it will go on like this. This is the way actually, through the anus, through the fecal matter. As I said to you, that the reason for the yellow color appearance of the fecal matter is even because of these two pigments, bilirubin and biliverdin. Who are these bilirubin and biliverdin? The detoxified forms of the heme, which are going to be combined with the bile juice. Okay, so that's the story here. That's remembered over. So that let me say once again the story of RBC from the beginning. As we said, the hemophoeitic stem cells will produce the myeloid stem cells. Not just no need to remember these particular issues here. Just for continuation, I added something here that for sequence for the continuation, I added all these things here, which are not there in the syllabus part. At least we have only one word, that is reticulocyte. Hemophoeitic stem cell, then they produce the myeloid stem cells, then retroerythroblast cell, then erythroblast cell, reticulocytes, and they produce RBC. So where all these are happening, that is in the red bone marrow. All these are the things are happening in the red bone marrow. In the red bone marrow, that all these things are happening. Okay, so whenever these particular red bone marrow produced this RBC, their lifespan, they contain only hemoglobin after the maturation, I said now. And then after all the 120 days, when they dies, what happens? When they dies, so that liver acts as the graveyard of RBC is the spleen and the globin. The spleen split the hemoglobin of RBC, dead RBC hemoglobin into heme and globin. And the globin is the protein part that will be recycled back again for the reproduction of the new RBC. But what about the heme? Heme is a toxic substance that should be eliminated. Then toxic substances will be sent to the liver, they detoxify the heme, and they detoxify in the form of two bile pigments. That bile pigments are said to be bilirubin and biliverdin. And these particular two pigments, along with the liver and gallbladder, also store the bile juice with these bilirubin and biliverdin and will be eliminated into the alimentary canal. So through the fecal matter. So that yellow colored appearance of the fecal matter is due to this. That's what the story, the total story of the RBC, about RBC. What is the function of RBC? So simple to say. RBC is for the transport of oxygen, the transport of oxygen. Okay. So that's it. And so let's take the item functions of all the blood cells, the functions of all the blood cells. So first of all, let's talk about the red blood cell. The red blood cells are in the transport of oxygen. The red blood cells are in the transport of oxygen, and they also little bit they participate in the transport of carbon dioxide that we are going to discuss in breathing and exchange of gases. So that's it. That is about the red blood cell. And next, let's take the cells one after another. The basophils, the function of basophils, the function of basophils. As we already said, the basophils are very less, 0.4%, the percentage we discussed, and the nucleus divided into irregular lobed, we said, and the cytoplasmic granules. We'll discuss about the cytoplasmic granules afterwards. So just only we want here the function. So did you hear this cell before anywhere? The basophil. I'll make you to remind. Mast cells. Did you hear the word, the cell, mast cell earlier?
Yes, sir.
Then you can say the function. As I said there, mast cell is nothing but basophil.
Produce heparin and histamine.
Yeah, inflammatory mediators they called as, and they release inflammatory the secretions that are the inflammatory initiators like the histamine, heparin, bradykinin, serotonin. That whatever the story we said about the particular mast cells earlier in the areolar connective tissue, the same we need to apply. That's all. So that's why we are not going to say anything here. Just simply we are going to say like the mast cells of areolar connective tissue, they are also, basophils are also doing the same. Histamine, bradykinin, serotonin, prostaglandins, platelet activating factor. If you remember, we said this as collar, dollar, rubor, tumor. Okay, so that we discussed that in the form of collar, dollar, rubor, tumor. So histamine, bradykinin, serotonin, prostaglandins, platelet activating factors, all we say inflammatory mediators, which are going to cause the inflammation, a process called inflammation. So that's about the function of basophils. Okay. Next, let's go for the eosinophils. Acidophils, we say. Eosinophils are acidophils. So eosinophils or acidophils. I'm not going actually in the traditional way. That means I made all the blood in the form of separate items: blood cells poiesis, blood cells pania, blood cells percentage, blood cells function. If you learn like that, that all the categorized form you learn, when we are reading the total text as a whole, it won't confuse us. Multiple issues are there, isn't it? That we discussed about the nucleus and we discussed about the the various issues when they increase, what you call eosinophilia. We use it to say eosinophilia. In general terms, they use it to say eosinophilia. They call the allergic response. Okay. So first of all, that function we need to see here. What they do actually, the function of eosinophils are, the function is they remove antigen antibody complexes. When a foreign body enters in the body, we know that we produce, foreign body can considered as antigen, and we produce antibodies. That antibodies, when they fight, when they bind with the antigens, that complexes, that we say that complex is the waste material, that complexes will be removed from the blood. They remove antigen and antibody complexes from the blood. The biggest function is that whatever the complexes which are rolling, which are moving in the blood, is going to be removed by eosinophils. But these are very famous in what form? These are famous in allergic reactions. You may heard over that word allergic eosinophilia. So when they increase it in number, it's going to be dangerous for us. They play a role in allergic reactions. Their number increase during allergic reactions. There are some other infections like helminth infections that these particular the eosinophils are going to play a role. That we know they are about only 2.3% they should present in our blood. If they increase, that's going to be danger for us. We call it as eosinophilia. Neutrophils. The next cells are neutrophils. The WBC, the granulocytes, we said three: eosinophil, basophil, and neutrophils. We already said they occupy 62% in the percentage of the whole WBC, and the nucleus is many lobe. That we discussed that also earlier, and the function. Now that we are taking the issue for function, they are active phagocytic cell. Neutrophils are phagocytic cell. They are the large in number, isn't it? That you may have seen over that acidophil with basophil, neutrophil. They are having the various types of nucleus. That we said in case of basophils, irregularly lobed nucleus is present, bilobed in the eosinophil, and neutrophils is with 2 to 5 lobe. So that's why the nucleus of these granulocytes is termed as polymorphonuclear leukocytes. These granulocytes, the polymorphonuclear leukocytes, acidophil, basophil, neutrophil. All the three can be considered as polymorphonuclear site, but neutrophils are about in large percentage in compare with the remaining, isn't it? 0.4% the basophil and 2.3 is only acidophil. So that's why this word is more pronounced for neutrophils. Okay, so that's why I'm writing here. All granulocytes can be called polymorphonuclear leukocytes. Okay, and as the main function we said already that because of their phagocytosis. What is phagocytosis? Eating, engulfing the microbial organisms. They act like like amoeba. They also have the pseudopodia. Then what they do, they engulf the microbial organism. These neutrophils are going to that if any foreign body is present, just imagine this is a foreign body. So these particular the foreign bodies are going to be engulfed by these neutrophils, and they do digestion. They destroy. So in that way, we say they are called microscopic policemen. The certain neutrophils here uh, considered as polymorpho nuclear leukocytes. Okay, so that's it. This is about neutrophils. Then in the same way, let's talk about the two. We have two agranulocytes. What are they? Lymphocytes and monocytes. Lymphocytes and monocytes. So particularly talking about the lymphocytes, if we are talking about lymphocytes, the lymphocytes we are going to discuss in detail in the coming chapters in immunology. If you remember, I already said about the percentage and nucleus. Nucleus is large spherical nucleus, percentage is about 30%. And function. Actually, there are two types of lymphocytes, T lymphocytes and B lymphocytes. Just to be remembered till the time when we go for immunology, the T lymphocytes and B lymphocytes. Okay, so these T lymphocytes and B lymphocytes, the T lymphocytes are responsible for that humoral immunity. T lymphocytes are responsible for humoral immunity. The T that, wait, we got a mistake here. That T lymphocytes are for cell mediated immunity. And the B lymphocytes are for humoral immunity. Details we'll discuss in immunology. So simply you need to remember the function as immunity. When microbe enters in the body, definitely we are going to kill. So how you're going to kill? Either with the T lymphocytes or either with the B lymphocytes. That's going to be the story. If at all B lymphocyte, we discussed little bit earlier. That B lymphocyte, we said they differentiate in the form of plasma cells, and that plasma cells produce antibodies, and that antibodies immunity, we call it as humoral immunity. It's enough up to here that because we are yet to learn more about this particular area. We have a separate chapter to learn this immunology. So that till the time you just remember this T lymphocyte cell mediated immunity. Okay, let's talk about the monocyte. Nothing to talk about monocyte. We already discussed about them. They're inactive. We call monocytes. When they become active, they are called macrophages. Function we discussed already. They are also like neutrophils. They are also in the phagocytosis. They are also in the phagocytosis. They are going to be settled down in different places of the body. These monocytes, when they become active, we call them as macrophages, are going to be settled down in the different places. Can I expect answer from you? In which place you have seen their settlement, that monocytes? Is there any place you have seen that they get settled down?
In
Histiocytes.
area.
Very
Histiocytes.
good. Monocyte. The story we learned earlier. They are monocytes. At what time? Inactive in the blood. When they become active, we call macrophages. And they settled in the areolar connective tissue, we call them as histiocytes.
Istos.
During migrating macrophages, we call wander.
Wandering
Wandering
macrophages.
Yeah.
macrophages.
So some more are there actually. They are yet to come. That they are yet to come. Like if they are present in the liver, we call Kupffer cells. In the brain, we say microglial cells. In the brain, we say microglial cells. And if they are in the bone, we call osteoclast cell. I think you got this. osteoclast. That in the bone here itself, in the first year itself, we got this, that osteoclast. They're all the descendants of monocytes, you need to remember. So, Inka, we have yet more to learn. In the brain, we called microglial cells. In the lungs, they are called alveolar cells. In the liver, they are called Kupffer cells. Wherever they go, the function is same. The single function. What is the function? Function is phagocytosis, engulfing the microbial organisms, those who entered in the body. So that we call say, we use it to say the phagocytosis. So that's it. It's all about the major issues of the blood we are seeing. And now it's a formality for us to go into the text, and we'll close this chapter in the text. We're going to close this chapter in the text here. So let me open the text for you. Structural organization in animals. Oh my God. Learned in the form of various items like percentage, cythemia, pania. So just a formality here. We'll make the completion of the blade. After the end, let us read the textbook here, how it is given. The fluid connective tissue or vascular tissue, is it to say. The fluid connective tissue include blood and lymph. The fiber-free fluid matrix called plasma and living cells, and that do not produce the matrix. It includes blood and lymph. It is a red colored. The blood is a red color, opaque and slightly alkaline fluid. The study of blood is called hematology. It is a tissue that circulates to various parts of the body through cardiovascular system. It is composed of blood plasma and formed elements or blood cells, RBC, WBC, and platelets. So here the plasma constitute about 55% and the cells are about 45% of the total volume of the blood. The total volume of the blood in an adult human being is about 5 to 6 liters. So need to focus on that calculations. So we discussed earlier that about the hematocrit value and a packed cell volume, the percentage of the total volume occupied by RBC when we are taken into a measuring test tube. Then we classified the blood into plasma and formed elements. So to be known in the plasma, the calculations 92% water and 8% of the solutes. It is the fluid matrix of the blood. Plasma is said to be the fluid matrix of the blood. It contains plasma proteins like albumin and globulin, and blood clotting proteins like the fibrinogen and prothrombin, anticoagulant material like heparin. Actually, they're all produced in the liver. And the globulins. Globulins which are formed in the liver and lymphoid organs are also going to be here only. Then albumin is the smallest and most abundant serum protein, and it is mainly responsible for the colloidal osmotic pressure of the blood. Yet to come, it will come in the next class. So what is colloidal osmotic pressure and how albumin is a savior to maintain the blood colloidal osmotic pressure. Okay, then fall in the levels of albumin in blood plasma results in edema. That also we'll discuss in the next class. The globulins are of the three types, alpha, beta, gamma. In that, gamma globulins are called antibodies or in immunoglobulins. And the function wise, we said the plasma as an acid base buffer, the pH is 7.4, and it also contains the salts like chlorides, bicarbonates, sulfates, and phosphates of sodium, potassium, calcium, and magnesium. And in the form of formed elements, when it comes, so we say they include erythrocytes, RBC, and leukocytes, WBC, platelets. The process of formation of blood cells is called hemophoesis or hematopoiesis. Total blood cells we call production, we say hemophoesis or hematopoiesis. As we discussed earlier, earliest stages of embryogenesis, blood cells from yolk sac mesoderm. Later on, the role is taken by liver and spleen as a temporary hemophoetic tissue. You need to remember, poiesis means production. That's why when you remember like that, they don't confuse all these kind of words. They can't be confuse you. In the final stage and the embryonic development of after birth and after the birth till our end of life, the red bone marrow is there in that work. The production of RBC. The production of RBC is called production of blood cells. Total blood cells we say hemophoesis, and the organ is responsible for that is called hemophoetic organ. And red blood cell. Remember erythro. Erythro, the word indicates the color red. Erythro means red. If you remember that in so many areas, in both botany and zoology, will be helpful for you in that manner. So erythro, the word indicates the red color. Erythrocyte, that means red blood cell. Erythrocytes of mammals. So just we learn this in the form of issues, isn't it? In the form of nucleus we learn. What is the condition of nucleus? Enucleate condition. So that in other mammals, the RBC shape may be elliptical in camels and llamas, but the human and almost all the mammals, except the camel and llama, that our RBC is biconcave and enucleate. That provides a large surface area to volume ratio. That the diameter wise, they are 7.8 millimicron in diameter. That next after that, the count wise, the number of RBC per 1 cubic millimeter, we said RBC count 5 billion in man, 4.5 billion in women. Next, decrease in the number of RBC called erythrocytopenia, which leads to the anemia. Then increase in the number of RBC is called polycythemia. When the RBC number is decreased over, the kidney respond and produce a hormone called erythropoietin. The vitamins which are required for the production and maturation of the RBC are said to be vitamin B12 and folic acid. Okay. So then after the mammalian RBC surrounded by plasma membrane, we call it as Donan's membrane. The nucleus and other cell organelles are lost in the reticulocyte stage. I just shown the full sequence there. That hemophoetic stem cells, that in that myeloid stem cell, proerythroblast cells, erythroblast cell, then reticulocyte, then they mature in the form of RBC. So they may have that cytoorganelle still reticulocyte stage, but after that they lose and become mature RBC. Mature RBC means without any protein, only contain hemoglobin, which heme contain 4 porphyrin rings and globin contain 4 polypeptide chain, which they can carry 4 molecules of oxygen. So finally, lifespan is 120 days, and that worn out RBC are destroyed in the spleen and liver. So that we discussed now, how they are going to be destroyed over. Yes, RBC of mammals are biconcave and enucleate. What is the advantage? Biconcave and enucleate. Biconcave advantage is our surface area is more for collection of oxygen. Okay, so then enucleate advantage, there is no nucleus and there is no cytoorganelles, means that is going to make us the filling the total content with hemoglobin. We can fill the total content with hemoglobin only. Okay, next. Let us see the white blood corpuscles. They are nucleate, they are colorless. They are colorless, very important. White blood cells are colorless. The complete cells, they are spherical or irregular in shape. Then capable of exhibiting amoeboid movement due to the extravascular areas. Diapedicis, this word also will come in the next class. They are larger than RBC in size and less than RBC in number. We learned their ratio is between them is 700:1 nearby. The count is 6,000 to 10,000 per cubic millimeter. The blood under normal condition. The production of WBC is called leukopoiesis, and the increase is called leukocythemia or leukocytosis. We can say that is in case of allergy cases. Then if uncontrollably they are dividing, we can say that is blood cancer. Normal increase, we say leukocytosis or leukocythemia. But if uncontrollable division is there, we call it as cancer, blood cancer, leukemia. Then decrease in the number, we call leukocytopenia. We are classifying the WBC into two, granulocytes and agranulocytes. The granulocytes are having the cytoplasmic granules. We call them as specific granules. that may take one of the three different types of stains. Do you know they are white blood cells? That means they are white in color. We can't see them in microscope. That means we need to apply staining on them. Staining means adding color to see in microscope. We need to add some color to watch the cells in a microscope. So some are going to be stained by acid stains. Some are going to take up only base chemical nature containing stains. Some are going to take up only that neutral stains. So based on that only, we call them as acidophils, basophils, and neutrophils. Staining material which we are using to watch them in under microscope. Based on that only, we classify them as acidophil, basophil, and neutrophil. In that way, we classify. Okay. Nucleus of the granulocyte is divided into lobes and assume different shapes. That's why we call them as polymorphonuclear leukocyte. As I said to you, it is more applicable for neutrophils. Okay. So based, because they are more in number in the granulocyte. So based on the staining properties, these are we classified them as three types: basophil, acidophil, and neutrophil. So then we completed all this in the form of tables. Just make a let's take a formality here. These are the basophils are the least common type and constitute percentage 0.4%, nucleus, irregular lobes, and cytoplasmic granules are fewer and irregular because they are granulocytes. That's why we need to talk about the granules also. So what the stain they take, basic stain, that's why we call basophil. And the function we discussed, we made the table that heparin, histamine, bradykinin, inflammatory mediators, they're going to be. They're like mast cells. Acidophils, percentage 2.3%, the nucleus, bilirubin nucleus, and so cytoplasm has large granules which stain with acidic dyes that such as eosin is a dye particularly. That's why we may call it as eosinophil. People pronounce it as isnophil, no matter, you can see that those who pronounce more, that will be the final generally. So they are in allergic reactions, their number increases during allergic reactions and Helminth infection, and they remove the antigen and antibody complexes in the form of function, we learned earlier. Then they are capable of phagocytosis and are not effective as neutrophils, but they are capable of phagocytosis. Next, neutrophils. The percentage 62%. The percentage of neutrophils is about 62%. Many lobe nuclei, 2 to 5 lobe. Cytoplasmic granules are small and abundant. And neutral dyes they're going to be stained with. They're active phagocytic cell, commonly described as microscopic policemen. Okay. So then actually neutrophils are having one more importance that they are having a bar body, we use it to say, drumstick body, that determination. That means in case of the female, the neutrophil is, if this is irregularly shaped neutrophil, the particularly in the female, there is a drumstick body is present over. Drumstick body, or say that heterochromatinized portion or bar body in the name of scientist Bar. He's identified that. So that bar body is present. So that indicates the presence, the determination aspect here. We'll discuss that in genetics. But you remember, there is a difference between the male and female in the form of neutrophils. If you say the neutrophils of the male, there is no difference of nucleus. 2 to 5 lobe nucleus is present, but this is the difference. This is called bar body. If bar body present, that is female. If this bar body or sex chromatin body or drumstick body, if it is not there, that is male. During embryonic development, the fetal prediction, that means the growing embryo, whether it is male or female, by staining this particular neutrophils, they use it to know. Details not at present required. Just we'll discuss in the genetics as well as in the reproduction topic. So agranulocytes. Agranulocytes, let's complete the story of them. That agranulocytes, they're about 30%. lymphocytes 5.3%, monocytes. We discussed about the lymphocyte. Their nucleus is large, spherical, with a little cytoplasm, scanty peripheral cytoplasm. And the lymphocytes are of two types, T lymphocyte and B lymphocyte. These B lymphocytes are going to produce the plasma cells. In further, they are going to produce antibodies for humoral immunity. And T lymphocytes are responsible for the cell-mediated immunity. The immunological reactions will be discussed in immunity. Where we stop here about lymphocytes, we start from there in immunology. Okay. Next, the monocytes. They constitute 5.3% percentage. The nucleus is kidney shaped and they are called macrophages. And their movements all over the body. We discuss like histiocyte, wandering macrophages, Kupffer cell, microglial cell. Wherever they go, they do one function. What is that? Phagocytosis. Next, the blood platelets. Count 250,000 to 450,000. They are formed from a giant megakaryocyte which is produced in the red bone marrow when it breaks down. When it breaks down, these particles are formed. We call them as platelets. That's not, they are not cells actually. They are a breakdown of a cell. A glass megakaryocyte broken down into many pieces, fragmentation. The average lifespan is about 5 to 9 days. They secrete a clotting material called thromboplastin, which play an important role in the blood clotting. They adhere to the damaged endothelial lining of capillaries and seal minor vascular openings, minor clotting we say. Okay, so that's it. It is all about the blood. So need to focus. Please frame the table like this. That when we are reading itself, you make the table. So when you frame the table, it will be helpful for us in that before we enter into the chapter reading, we'll search and we'll go only in the table form. That means we are learning percentage, we are learning cythemia, we are learning pania. If you learn like that separately, the types of nucleus in all the blood cells, the percentage in all the blood cells, the count in all the blood cells, the poiesis, production, how it is named as, cythemia, how it is named as, pania, how it is named as, and the function of the blood cell. If you learn like that when you are reading the textbook, you feel smooth. So you people need to do this. That's why these blanks are empty. These particular table, which is always empty like this, that a student should do here while reading the textbook and should focus on these particular tables. You may get this directly as in the form of math the following in examination. So that's why I request all of you to make the table, and tomorrow we continue with lymph. The lymph is the topic here that we discuss with, we'll go with the lymph in the next class. Okay.